Gram-positive cocci. Cocci are spherical or oval-shaped organisms. Staphylococci looks like bunch of grapes. Gram-positive cocci arranged in clusters. Why they are arranged in cluster? Because they divide in whole like this. Everybody, they divide in all planes. Daughter cells fail to separate out from mother cells. That's why they are arranged in clusters. They look like bunch of grapes. Gram-positive cocci arranged in clusters. Staphylo means grapes. Staphylo means grapes. They are gram-positive cocci arranged in clusters. They are gram-positive cocci arranged in clusters. They are gram-positive cocci arranged in clusters. Streptococci, cell division takes place in single plane, not in all other planes. And daughter cells fail to separate out from mother cells. It is called as it's serpent. It looks like serpent. Gram-positive cocci arranged in chain. Gram-positive cocci arranged in chain. Gram-positive cocci arranged in chain. That is streptococci. Then pneumococci. First two cocci, one is bunch, one is grapes, another is serpent. Next three cocci are diplococci. Next three cocci are diplococci. They are in pairs. They are in pairs. They are in pairs. The next cocci is pneumococci. It's called pneumococci because it gives rise to pneumonia, consolidation of lung. It gives rise to consolidation of lungs, pneumococci. It is lancet shaped, flame shaped, flame shaped, broader and facing each other. It's lancet shaped, flame shaped, and there is a two cocci enclosed in a single capsule. Two cocci enclosed in a single capsule. It's a lancet shape. It is lancet shaped, two cocci enclosed in a single capsule. It's also gram positive. Gram positive. Lancet shaped. Or flame shaped. Diplococci enclosed in capsule. Enclosed in capsule. Gram positive, lancet shaped or flame shaped diplococci enclosed in capsule. Gram positive, diplococci. The first three cocci are gram positive. Next is gram negative. One is they predominantly, most of them are intracellular inside the neutrophils. One is plano-convex. Half moon or plano-convex. Half moon or meningococci. They are inside the pus cells. Inside the pus cells. The next one is kidney shaped. They are kidney shaped. concave end facing each other. Concave end facing each other. They are kidney shaped, concave end facing each other. They are kidney shaped. First two are gram positive. First three are gram positive. Next two are gram negative. So repeat with me. Staphylococci looks like bunch of grapes because they divide in all planes and daughter cells fail to separate out from other cells. Streptococci arranged in chain looks like serpent because it divides in single plane. Daughter cells fail to separate out from other cells. Pneumococci, lancet shaped or flame shaped, broader and facing each other. Two cocci enclosed in a single capsule. Two cocci enclosed in a single capsule. The next two cocci are gram negative. First one is plano-convex or half moon. Plano-convex, half moon. Everybody. Plano-convex, half moon shaped, uh, plane surface facing each other. The next one is kidney shaped, concave end facing each other. Kidney shaped, concave end each other. First, staph, strepto, and pneumo are gram positive. Staph, staphylococci, streptococci, pneumococci are gram positive. Staphylococci, streptococci, pneumococci are gram positive. Gonococci, meningococci are gram negative. Staphylococci and streptococci are also called as pyogenic bacteria because they produce pus. Staphylococci also produce non-pus producing lesions due to toxin. The three non-toxin producing lesions are food poisoning, food poisoning, toxic shock syndrome, toxic shock syndrome, superficial skin scalded 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 syndrome. Just 1 minute. Superficial, just repeat all the things what I said. Staphylococci, forgotten my phone on my table. Very important. Streptococci also produces type 3 immune complex mediated infections. There are two. One is acute rheumatic fever, then acute glomerulonephritis. Acute rheumatic fever and acute glomerulonephritis. Acute rheumatic fever and acute glomerulonephritis. Acute rheumatic fever and acute glomerulonephritis. This is due to deposition of immune complex on heart wall and glomerular filtration membrane. Streptococci is also called as flesh-eating bacteria. It gives rise to infection called necrotizing fasciitis. It's nicknamed as licks the joint and bites the heart. Licks the joint and bites the heart. It gives rise to acute rheumatic carditis and acute rheumatic arthritis. Now moving on to bacilli. Bacilli, find out where is the duster. Bacilli are classified into three criteria. Bacilli are classified into three criteria. One is Gram reaction. The second one is spore forming capacity. Third one is acid fastness. Write down. One is spore forming capacity. Spore bearers are ability to form spores. Ability to form spores. Ability to form spores. The second one is acid fastness. They do not get decolorized with acid alcohol mixture. They do not get decolorized with acid alcohol mixture. Third one is gram staining. Third one is gram staining. Bacilli are rod-shaped organisms, 3 micron in length and 0.3 micron in breadth. 0.3 micron in length and 0.3 micron in breadth. So now they are classified depending on three criteria. Spores. Spores are highly resistant. Everybody, spores are highly resistant resting stage of bacteria. They are formed during unfavorable conditions. They resist desiccation, drying, high temperature. They are important in sterilization. They are killed only by autoclaving or hot air oven. What is the temperature of autoclave? 121 degrees, 15-pound pressure, 21 minutes. 15-pound pressure, 21 minutes. Hot air one, 160 degrees for 1 hour and 180 degrees for half an hour. So important one is the anthrax bacilli. Anthrax bacilli. Anthrax bacilli. It divides again like streptococci in single plane. It gives rise to bamboo stick appearance. 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 Next one is Clostridium. Clostridium. So there are Clostridium tetani, Clostridium welchi, Clostridium welchi, Clostridium botulinum. Clostridium tetani, Clostridium welchi, and Clostridium botulinum. Clostridium tetani, Clostridium welchi, and Clostridium botulinum. And it has got tetani has got terminal bulging spore. It gives rise to drumstick appearance. Drumstick is not your vegetable drumstick with which you hit the drumstick appearance. Clostridium tetani gives rise to tetanus. Clostridium tetani gives rise to tetanus. Clostridium welchi gives rise to gas gangrene. Clostridium welchi gives rise to gas gangrene. Clostridium botulinum gives rise to deadliest food poisoning, and this is used for one of the biological welfare. Now moving on to the acid fast organism. Stained by Zlinson staining. One of the exercises is given in your practical exercises. You have to do Zlinson staining and gram staining both. You have to do Zlinson staining and gram staining. So remember the steps in gram staining. Violet, put violet, gentian violet, put iodine as a mordant, and we put alcohol as decolorizing it, dilute carbamazepine. There is a counter stain. One is a primary stain, second one is a counter stain. This is a mordant, this is the decolorizing agent. So whereas Zlinson stain is 3 steps. Remember it as CAB, CAB. Cover the smear with concentrated carbamazepine. Heat it intermittently from beneath with a spirit lamp, taking care not to char, black or break the slide. Taking care not to blacken or char or break the slide. And decolorize with 20% sulfuric acid or hydrochloric acid. Counter stain with, here please remember there this is concentrated carbamazepine, here it is dilute carbamazepine. There it is concentrated carbamazepine. Decolorize with 20% sulfuric acid and counter stain with methylene blue. Counter stain with methylene blue. The example of acid fast organisms are Mycobacterium tuberculosis, Mycobacterium tuberculosis, and Mycobacterium leprae. Mycobacterium tuberculosis and Mycobacterium leprae. Mycobacterium tuberculosis and Mycobacterium leprae. Mycobacterium tuberculosis, Mycobacterium leprae. So these are as the third criteria for classification of bacilli is gram staining. Some bacilli are gram positive, some bacilli are gram negative. Gram positive bacilli which look violet in color. Corynebacterium diphtheria. Corynebacterium diphtheria. Corony means thorn like, hold like this. Hold like this. Thorn. Thorn. Because they divide like a stem or shoot of the tree. They divide like stem or shoot of the tree, and daughter cells fail to separate out from the mother cell. Corynebacterium diphtheria. Diphtheros means they give rise to leathery membrane in the throat. They give rise to leathery membrane in the throat. They give rise to leathery membrane in the throat. Next one is Lactobacillin. They are present in the milk, that's why it's called as Lactobacillin. Then Listeria monocytogen. Gives rise to meningitis. Listeria monocytogen, it gives rise to meningitis. Then Erysipelathrix. Erysipelathrix rosopathiae, it gives rise to skin erythemata. Important organism you should know is Corynebacterium diphtheria. Rest are all Lactobacilli, Listeria, Erysipelathrix. These are all gram positive. Now moving on to the gram negative bacilli. Moving on to the gram negative bacilli. Moving on to the gram negative bacilli. They look pink in color, whereas pink or purple. Gram positive look violet in color. Gram positive look violet in color. They all belong to Enterobacteriaceae. They all belong to Enterobacteriaceae. The differential medium, selective medium used for growing McConkey's medium is medium called McConkey's medium. McConkey's medium. So it consists of plant. Remember plant. Peptone, lactose, agar, neutral red, tarocolate, peptone, lactose, agar, neutral red, and tarocolate. Peptone, agar, tarocolate, and neutral red. So depending on the fermentation of lactose, they are classified into lactose fermenter. They produce pink colored colonies, and non-lactose fermenters they produce pale or colorless colonies. Pale or colorless. Example of lactose fermenter colonies are E. coli and Klebsiella. 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 Non-lactose fermenters are Salmonella, Shigella, Proteus, Pseudomonas, and even including Vibrio cholerae, although it will not come under Vibrio cholerae. Salmonella, Shigella, Proteus, and Pseudomonas. Okay, these are all example of gram-negative bacilli. So next class we will take the comma-shaped organism. Comma-shaped organism is Vibrio cholerae. They look like, and it is motile with the monotrachate flagellum. Comma-shaped organism is cholera bacillus. Okay, thank you very much.